So our next speakers are Mats Tolborg and Rupert Foch from the James Hutton Institute in Scotland, introducing us to quantitative risk analysis. Thank you.
Yeah, so yeah, I should maybe start to begin with that. I don't really work on AMR things, so this is more like a general introduction to quantitative risk assessment of chemical exposures and that exposures to pathogens. So my plan was just to give a general introduction and then go through an example of a quantitative risk assessment that Rupert and I did a few years back, which was on using recycled wastewater for different end-use purposes, and then finish with a slide on what the challenges in doing such a risk assessment for AMR-related problems might be. And we're maybe hoping to open up the discussion at that point, because we think that's where people might have some interesting inputs. Okay, so what is risk? I think this is one of these things, depends who you ask. In general, I think a general definition is that it's the probability of suffering harm from a hazard. And it's often described through the probability of a hazardous event occurring multiplied by the consequence if this hazard is occurring. So that's what's illustrated on this figure here. And what you actually see with lots of hazards is that they kind of fall on a curve like this, that the hazardous events that tend to happen frequently are also the ones that normally have fairly low consequences, whereas the catastrophic type of risk events, like Earth being hit by a massive asteroid, they luckily don't happen that often. And obviously, we are concerned with high risk events, the ones with high probability of happening and high consequence. Let me just and risk management is all about sort of do different measures to sort of move a hazardous event from a high risk event to a low risk event. So the definition of risk that we're using in this talk is that it's the probability of harmful effects to human health, or it could be ecosystems as well, resulting from exposure to an environmental stressor. And here in this context, the stressors could be chemicals or microbial, it could also be physical or radiation and things like that. So when you do quantitative risk assessments, usually you base this on a source pathway receptor approach. You often have a source of contamination, and you have some receptors that you're concerned with protecting. That could be human health, for example, and then you have the pathway that connects the two. And only if there's a full pathway linkage between the source and the receptor is where you will have risk, basically. So in practice, these risk assessments, they involve 5 steps. So there's a planning and scoping phase, there's a hazard identification, there's an exposure assessment, and then there's a toxicity or dose response assessment. And finally, you summarize it all in a risk characterization. I was just going to go through each of these steps, sort of in what they involve in general. So the planning and scoping stages, I mean, it's kind of what you would do almost in any kind of modeling exercise. You try and frame the problem that you're looking at. So this is where you ask a lot of questions such as who, what, and where is at risk. So you're defining your receptors. So that could be you're looking at general population, individuals, or it could be subgroups, like particularly exposed subgroups, vulnerable subgroups, like children or pregnant women. Then define what are the environmental hazards of concern. Are you looking at chemicals? Are you looking at pathogens and so on? And also looking at whether you're looking at individual single hazards or multiple hazards as well. It could be a mixture of things. And then you look into where do the hazards come from, what are the sources, and how does exposure occur. So that's where you start defining the pathways and routes of how a chemical can make its way from the source to a receptor. You're also looking at what are the health effects if this happens. Is it like an acute or chronic type of health effect? And based on all this, you basically formulate an initial conceptual source pathway receptor model. The hazard identification step is more sort of a detailed characterization of the sources and the hazards that you're focusing on. So this is where you start looking into what are the hazards properties, how do they behave if they make it into the environment, what kind of concentrations do we have in the sources, and what are the emissions. And here it might, in some cases, be necessary to be a bit selective about what hazards you're looking at. You might want to select so-called reference hazards if you're looking at very complex sources like, for example, wastewater, which contains lots of different types of chemicals and bacteria, pathogens, and so on. It's often not feasible to try and do a risk assessment for all of them. So you'd be trying to select those that have high occurrence, are particularly toxic, or persistent in the environment. You also want to then characterize what are the concentrations or concentration in the sources, and you can have quite a degree of variability. So that's an important thing to characterize as well as part of this step. Then there's the exposure assessment. So the ultimate aim of that is to estimate what is the dose of the exposure concentration that a receptor will be exposed to. So you're concerned about what is the magnitude, the frequency, and the duration of the exposure to an agent in the environment. Sometimes this can be measured directly, but very often this assessment it's based on some kind of transport and fate modeling. So how does the contaminant actually make it from the source to the receptor? Is there any kind of environmental attenuation going on, and what barriers does it have to pass to make its way? This obviously depends on the kind of pathway and route. So pathway by pathway, I mean, that is actually the route, the pathway takes from the source to the receptor, whereas the exposure route is how does it make its way into the receptor. So if we're talking human health, are you ingesting it with your food, or are you inhaling it, or are you even dermal contact is a possible route. And also it depends, of course, on the type of receptor we're looking at. The kind of things you estimate typically for if we're looking at human health and the risk of to human health from chemical exposure is this thing called an average daily dose, which is calculated using this expression here. So it's fairly simple. It's the concentration of your hazard in whatever media that the human might ingest. So that could be for contaminated food crop, or it could be contaminated water. Then it's the uptake or intake rate of that specific media. And then you always sort of report it per kilo body weight. And then you have this fractional time of exposure. That's just sort of an averaging time, if you like, is how much contaminated food do you eat over the time period concerned, and so on. Then next step is the dose response assessment. So Jan talked a bit about this. So basically that's just to look into at what kind of dose do we start seeing effects on human health, for example. And the aim of that, I would say, is typically to try and estimate what is an acceptable level of exposure. That is like when, what is a safe level. And the way this is derived is typically from dose response data, or if you're looking at pathogens, it can also be from outbreak data in populations. So if it's from dose response data, it's basically just like you do some experiments on like algae or mice or whatever, and then you look at what kind of concentration or dose do you start seeing effects to the population. Yeah, people might have heard of the lethal dose 50 concentration or dose. But there's also other measures such as the lowest observed effect level and the no observed concentration level. And these measures are then often used to derive safe doses for example, for human health. This is a reference dose. So the way people do that is that they, for example, use the no observed effect concentration in mice, and then to account for the fact that the humans are not mice and they are a bit, you want to be protective of human health, you divide that level with some uncertainty factors. And they're a little bit controversial, I think, to say the least, but it's basically a precautionary way of trying to extrapolate concentrations from one organism to another. It's the same if you're doing ecological type of risk assessment, then you're concerned with the predicted no effect concentration. And again, it's very common to take the no observed effect concentration and just divide it with some kind of assessment factor, which accounts for the fact that that this is done under controlled experimental conditions, and it might not be the case in the field and so on. So this is more for chemical exposures. For bacteria, for pathogens, it's the dose response is more sort of based on dose response models and looking into the what dose results in certain probabilities of infection. So there's a number of models that have been fitted to outbreak data or to control experiments to predict the probability of infection for various doses. So there's a slight difference depending on what hazard you're looking at. And then the final step is this risk characterization. So in sort of very simple terms, this is just where you're comparing the dose that you've estimated in your exposure assessment to the acceptable exposure levels from your dose response assessment. So, and what you normally do is you just calculate a so-called hazard quotient or risk quotient by dividing the dose with the reference dose. And if that's above 1, that's where you start having a risk. And if it's below 1, then you consider it to be safe. And that's, in a way, sort of in very simple terms, what you do. It's also where you would normally start characterizing the uncertainties in your assessment. So these kind of risk assessment, they rely on a lot of assumptions and model inputs that are generally very uncertain. And it's obviously important to address these uncertainties to enhance stakeholder confidence. You do that by just being clear and open about what are the limitations in the approach you're using and the data gaps, and just basically communicate the uncertainties. And there's various ways you can address the uncertainties. I'd say the most common way is almost just to use a very sort of precautionary approach. So all inputs that you're using in your assessment, you just tend to use worst case or near worst case assumptions. You can, if you have like a model, you can do like sensitivity analysis to look more closer into what are the inputs that are really driving the risks. And then you can do these type of uncertainty propagation methods. It's illustrated here. Some people might be familiar with approaches like Monte Carlo simulation, where instead of having one value for your input, you specify a probability distribution for your inputs, and then you propagate that through your model, and then your output also becomes a probability distribution, which then characterizes the uncertainty in your estimate. So I was just going to go through an example where we've used a quantitative risk assessment, and as I said, this was to do with looking at the risk associated with using reclaimed water, recycled water from wastewater treatment plants for different irrigation purposes. So again, this was then based on the source pathway approach. So the source in this case was the final effluent from wastewater treatment plants. We're looking at exposure pathways that were using the water for irrigating for amenity purposes. So that would be like using to irrigate public parks or golf courses and things like that, but also use it to irrigate food crops. And the different carrier media you would consider in these scenarios, if you like, is soil, water, and food crops. And the receptor we were considering here was human health and what were the health risks if ingesting media that's been contaminated with final effluent. So in this particular study, we were actually interested in trying to work out what concentration levels would be safe at the source to use for irrigation purposes. So we ended up doing like reversing this source pathway receptor model. So we started at the point of what is a safe dose at the receptor point, and then reversed the pathway modeling to say what is the corresponding safe concentration in the effluent, and that would then give us an idea of how much should the water be treated at the wastewater treatment plant for it to be safe. So this is an example where like risk assessment can be useful for setting standards, water quality standards. So the hazard identification here, that was we did a huge review of what are the potential harmful agents in final effluent. So this was one of these examples where it's like absolutely hundreds. So that was a challenge. And in the end, we were considering 3 groups of hazardous agents. So we were looking at microbial hazards, a selected number of pathogens. We're looking at potentially toxic elements. So this was some heavy metals, and then also some organic contaminants. For each of these, we did a review of what are the key properties of these different hazards. So there's stuff about how do they decay in the environment. These factors here is about how do they adsorb to the soil, for example. There's like how do they yeah, well, there's also about like how toxic are they, but I'll come back to that later. So for the exposure assessment, as I said, this is about trying to estimate what's the dose that a receptor is being exposed to for each of these exposure pathways and routes. So we're looking at two end-use scenarios, as I was saying. So one was the amenity irrigation, and we're considering that humans could be exposed to chemicals or pathogens in the effluent directly by accidentally ingesting effluent after it had been applied to land, but also ingesting soil that had been irrigated with effluent. And for the food crop irrigation, we had similar pathway, but obviously also ingesting crops that had been grown on land irrigated with effluent. So this also shows that to do these calculations, we needed to estimate what would the resulting concentration be in the soil as well as in food crops to calculate what is the likely exposure to human health. And to do that, we set up a fairly simple mass balance model for a soil volume that had been irrigated with effluent. So it's basically just a mass balance, and that considers the fact that the hazard might adsorb to soil material once it's entering the soil volume, and that it might experience decay over time. And the uptake by plant we model using simple bioconcentration factors, which is just you can find published relationship between what's the concentration in plant if you have certain concentration in soil. So it's a pretty simple way of doing that. For the dose response or toxicity assessment, again, the aim is to estimate safe doses. In this context here, we were able to find, for the chemicals, we were able to find reference doses in the literature for the heavy metals and the organic contaminants. And for the pathogens, we found, we ended up using some data from like models that had been fitted to dose response data in the literature, and there's a so-called fractional Poisson model we're using for that. And that gives you the probability of infection. And then to calculate a safe level, we are using, this is kind of a common assumption that an acceptable annual risk of infection is 1 infection in 10,000 people per year. So that was kind of what we used to then calculate what's an acceptable dose of pathogens. Oh, and yeah, I should say that, so we used all this to try and calculate what the safe concentration in effluents would be to have acceptable risk. I say one thing we're looking into in this study as well was how to quantify uncertainties. So as you can see in this table here, a lot of the input parameters, we've defined a best estimate value for all of these, but also a range, an uncertainty range. And you can see a lot of these input values are very uncertain or can easily range like an order of magnitude. So we're looking into how that could influence the results. And in this study, we used an approach called fuzzy logic to propagate uncertainties through the results. So it's a little bit similar to Monte Carlo simulation, but Monte Carlo simulation is a probabilistic approach, whereas this fuzzy logic is a possibilistic approach. So it almost looks at what, giving a range in your input parameters, it calculates what is the worst and the best case result, giving that range. So just like this is called the membership functions of the result. And we have, if we use the best estimate of all the input values, then we have this result like at the pointy bit of the curves. Whereas if we use the extreme values for the whole range of all the input values, then this is the range in the output results we, for example, get for cryptosporidium, and this is the range we then get for naphthalene. And we just see that we get like several orders of magnitude uncertainty in the results. So it was just one example that was demonstrating how uncertainty influences the results. So that was kind of it I was going to say about the quantitative risk assessment, but then actually kind of wanting to just talk about what are the challenges in doing a quantitative risk assessment of the antimicrobial resistance. And I mean, if we were to use this kind of framework with the hazard identification, exposure assessment, dose response, and risk characterization, maybe put some questions in that we could potentially discuss. So what are the main hazards of concern when we look at this? And again, what are the key routes for environmental EMR for the exposure assessment? And do we need, I think it would be interesting, like following on from Jan's talk, do we need, like how complex models do we need to model the transport and fate in the environment and quantify exposure? The dose response assessment, what is it exactly we want to assess here? And for the risk characterization, that could be quite interesting to discuss again, what they actually constitute an unacceptable level of risk when we talk AMR. So I don't know if it's possible just to open the discussion. I don't know, Rupert, you might also want to add things to this at this point. I mean, I can say we added a few things that we've been discussing under each of these steps, but we thought maybe we could have people could have come with some inputs first, and then we could show what we've been thinking afterwards. I don't know if that's an approach.
Well, we can try it. So
also if there's any questions.
Any comments on that or inputs?
Hi, Mats. I have a question. Thank you for the talk. I really like it. Less equations so I can follow easier for me. So my main question when I see all these models is, for instance, how do you calculate what is the exposure rate, for instance, when you have this part of the golf camp, no? Like, how do you calculate what is the ingestion of water from the sprinkles or the ingestion of soil? Where do you get this data? Is people doing this kind of studies apart and then you just take the data, or you need to do it yourself and take the data yourself?
That's a good question. And actually, I totally forgot to mention this. Obviously, that's quite a key input for the exposure assessment, but there's all sorts of studies being done on how much soil does children eat every year and how much food crop do people eat and so on. And you can find so-called exposure handbooks that's been published with rates of all sorts of things. So there's actually quite a lot of published studies. I guess it also, again, depends a bit on the context. So whether you're looking at a specific area, then you would want kind of ingestion rates that are specific to that area, or whether you're looking at it more sort of generically or at a sort of wider scale.
I mean, certainly the exposure factors handbook, it's an American publication, but it provides information on how much time people spend doing all sorts of different activities, and it's split by age, by gender, by ethnicity, blah, blah. There's no equivalent publication for any other part of the world that I'm aware of, but often we use that one because it's so detailed and so comprehensive. If you were studying an actual population, you might take the time with, say, questionnaires or whatever to find out for the key exposure routes what those people are actually doing. But when it comes to doing it in a generic or theoretical way, we often use publications like the Exposure Factors Handbook, and maybe adjust them a bit because the American population, we know it differs in certain ways, say, compared to European or Asian or whatever. But that's often the starting point.
Yeah, because I was very surprised, like the part of the crops, I can understand that there is data of ingestion and everything, and I also understand about the kids and the playgrounds, but this specific part that you were talking about, the golf camps, like I was thinking, like, have they done a study of how much soil do they ingest in a golf camp? This is super specific. So I was really like, do you do this kind of studies? And like I was wondering, like, if there is a very specific data on that, or you just indeed need to extrapolate from other things.
There's data on how much time people spend on golf course by age and gender. There isn't the specific data on how much soil people ingest, again, by age, gender, etc. There isn't the two combined, so you have to make assumptions of how you combine the two. If you see what I mean. But there is specific data on both those aspects. Yeah.
Okay. Yes, I was curious about this specific example because, yeah.
I was going to ask, Mads, for this project on AMR, would instead of sort of dose response curves, are we more trying to look at the likelihood that we are within the selective window of antibody concentrations? So, where there's this range of antibody concentrations that the sort of, bookend that selective window. And so the likelihood that we are within that, basically. But I feel like that we would need some data, much more data on the relationship between the flow and the concentration of the antibiotics.
Yeah, I mean, that's a good question. I'm not really sure I know yet. And is that if we are looking at it from the water point of view or from human exposure? Because it's one of these things that, like, especially with the dose response assessment, like, you can look at the probability of being infected, but it should matter whether you're being infected by a resistant bacteria or a non-resistant bacteria, because it's sort of impacts whether it's treatable or not as well. So that's maybe one thing that's worth considering. I know that in some studies and risk assessment studies, instead of just having sort of a safe dose, they use so-called DALYs, which is disability adjusted life years, I think it stands for. So that accounts for not just that you're being infected, but it also accounts for the severity of the infection and how long you might live with the disease. And it actually enables a comparison of chemical and pathogen exposures. That's maybe a way you could account for some of these things. I'm not quite sure, to be honest.
Yeah, how if you wanted to go to infection risk, how do you move from the dose response type curves to infection risk? Is that, because this is much more like an epidemiological model, right, with caseloads and density of human contact and things like this?
So how they do this in practice? with the.
Or it's just, yeah, wondering how we would implement that kind of thing. How would we go from a dose response type to a more epidemiological infection risk?
Yeah, I mean, the published data, they'll be for specific organisms, and they usually come from specific outbreaks. So I mean, the famous one for E. coli 0157 is a scout camp. I remember that as a scout camp because it took place in northeast Scotland. So there they've studied a particular outbreak. They have some idea from measurements they've taken, I guess, at the site from what people got on their hands. I think that particular outbreak was from animal feces, I think, where they camped, like sheep manure or something. They make some estimate of the exposure and work out from that the probability of infection, because some of the people get exposed and get infected. Some, you know, they show symptoms, etc. Some of them don't, and you get these data sets which are the best things you've got. They're not brilliant. You can already see the huge uncertainties in trying to estimate that. So yeah, that epidemiologic data is then used. So for a particular dose, you get this probability of showing symptomatic outcome. I wouldn't necessarily call it infection, because obviously you can be infected and have no symptoms, but you know, it's a probability of actually being sick. So you have that information for some organisms in some settings, and is often used widely then in all sorts of settings, that those particular data. So the one I was thinking of, which gives you a beta Poisson model of infection for E. coli, is used all over almost. It becomes almost a standard of what is used as the best thing available. Whether that is right or wrong is another scientific debate, but that's often ends up being used. So you know, you read off that curve, the exposure is this, so the probability of infection is that. I think what Mess was talking about with looking at presenting results with error propagation helps take into account some of these uncertainties that are inherent in these data sets, and actually try to present them in a more explicit way, because often in an assessment of this nature, all that uncertainty is just hidden. You know, people will select the worst case and just go with the worst case, assume the exposure was very high and go with that. Yeah, I'm not sure I'm answering your question though, Josh.
Yeah, Rupert, this is related to the question in the chat from Kiri Rogers, who is asking, from experience, what is your opinion on the validity of results when having to make numerous assumptions, particularly when we would be looking at such a complex and unexplored avenue of AMR? So I guess this question fits in here directly. You know, you have mentioned the importance of using error propagation to work out how much uncertainties in the assumption feed into uncertainties in the final risk analysis. And I think that's also generally true for all models, that you need to explore how much your results would be affected if assumptions were wrong, or if you're changing assumptions. So this is called sensitivity analysis, and is a crucial part of this, so that you get a good understanding of whether the assumptions you're making, which may be wrong, or sometimes you simply don't know how good they are, whether they will be affecting your conclusions or not. Some assumptions have little effect. Other assumptions can be critical, and if some assumptions are critical and you do not have reliable evidence for them, then you know that you need to know more, and you have to be very careful with your conclusions. But this is always the case in science. You know, we work with very complex environmental systems, where also any measurements come with lots of caveats, because you only take a sample at a certain time of the day in a certain place, and you don't know how much it would be different at night. You know, so uncertainties are everywhere, and the modeling does actually helps you to identify how big the problem is of having uncertainties in certain assumptions.
Yeah, and I mean, certainly with the risk analysis, our aim isn't necessarily to get the correct answer. It's to provide a steer. It's to provide the best analysis that we can, that hopefully provides a steer on how you might deal with those hazards, how you might manage them. Often when we do some of these larger risk assessments, larger generalized ones, they often involve many stakeholders, and all the assumptions would be argued between the stakeholders and agreed in a room. So although we know that every single assumption has uncertainty attached to it, you come to a consensus on what we feel is appropriate, and then the analysis should hopefully people feel it guides their decision making. You know, if it says this activity is more risky than that activity, it guides that decision-making process. So we don't have to, I suppose, come up with the perfect answer, but it needs to be an appropriate answer and as close to the true answer as we can possibly get, sort of thing, if you see what I mean. Yeah. But it's a very important point. All modeling exercises are full of assumptions, and the assumptions need to be challenged and scrutinized at each step. And if we come back at a later date and we can, you know, reduce the uncertainty somewhere, that's also great. You never see a model as a fixed and final answer.
Yeah, there is a question from Emma Travis in the chat. Do you want to address this, Mats or Rupert? You see it?
Yeah.
Yeah. I think it's a distinction between resistant bacteria that are present because they come into the system from the sewage, and resistant bacteria that have been selected in the environment, or selected for resistance in the environment because of the presence of antibiotics at too high concentrations. As a separate pathway, or adding them up. I mean, two different causes feeding into one concentration of resistant bacteria, I guess.
Yeah, I suppose my question is the exposure the same? As in, are you being exposed to the same thing when the human ingests it, or is the human being exposed to two different things? Because that would to me make the distinction. So if the organisms were different, that would obviously be two different hazards you're assessing there. But if it's the same organisms with the same resistance, that would be the same. in my view, anyway.
Yeah. Is this related to how, like, what happens in the pathway? So from the source to the receptor, I suppose.
Interesting. So this is Emma here. I think that the key thing here is that you get the ingress of microbes, which might be restricted through a different method into the environment compared to the influx of, for instance, the antimicrobials, which would then select. And as you say, that might be in the same organism, or you might also get a transfer of the genes between different organisms. So you might have different organisms. And I think that's probably quite important to consider when you're looking at how to control the risk, because, for instance, in the UK, a lot of the bacteria will be going into the rivers, for instance, from wastewater treatment when there is an excess of rain, and it won't be going through the normal thing. You'll get this sort of flow through, which is probably responsible for a lot of the microbes actually in the river. And that's a different process to stripping out the chemicals, for instance, so antimicrobials plus other ones which might actually co-select for the microbes as well. So I think that it's quite an interesting one to consider. I don't know what your thoughts are. So it's sort of both the end game might be different, but also the input might differ in terms of mechanism of restricting what goes into the environment.
Yeah, and I think in the study sites and in our project, I think some of the effluent is treated to some extent, and some of the effluent isn't. It's directly discharged. So you get those two, a bit like what you're talking about, Emma, in the high rainfall situation. You get that two different streams of input, and they have different characteristics. So I think it is a situation we need to get some sort of handle on. Yeah.
Yeah, it's definitely a bit more challenging than the stuff we've done on like chemicals, because you don't have all that gene transfer stuff you need to consider as well, which I guess will impact what eventually arrives at the point of receptor. So there's definitely some challenges there for risk assessment anyway. And I think often, like the calculation, as you can maybe see from this slide, is often more sort of back of the envelope type of calculation you end up doing to get sort of rough estimates of how things might be. I mean, in principle, you could, I guess, if you had great data and knowledge, you could use more complex models like what Jan presented. Or, I mean, I personally think it would be interesting to look at something like what Jan has presented, but whether you could simplify it so that you could say under given conditions, say if we know it's an aerobic system and temperature ranges like this, can we then say something about if this is the type of source we have, can we then simplify the system and the exposure assessment, or do we have to account for all the more sort of detailed processes about growth and die-offs and gene transfer and all that happens during the pathway?
Yeah, I think that's interesting, and I think that it might be that that it could potentially be simplified if it looks like one of these risks completely outswamps the other, such as when you do get this untreated effluent with rainfall, for instance. And I think that it's the relative risk is very much then from your microbes, for instance. So I think that's interesting. Thanks very much for that.
So Chiara's asking about the PNEX, and that's what I was asking about as well. So these are multiple nearly all, I think all of our sites will have super complex mixtures of toxic compounds, particularly antibiotics. So my thinking is that you take the one that's most toxic at the highest concentration as your sort of PNEC because then we're looking at a PNEC for organisms in the environment, as well as a PNEC for humans, I suppose, for an antibiotic rather than a bacterium or a combination of bacteria. So because I think Chiara's worried about the multiplicity of compounds. It makes a very complicated picture.
Yeah, I mean, what the modeling of the selective window is that we are doing is trying to address this question, because the PNEC concentrations, so predicted no effect concentrations used for antimicrobials in the environment, are based on the MIC values of the compounds. And
you mean the Larson ones, the ones that Will developed, Larson and Will? Yeah.
Yeah, so basically, you look at the distribution of MIC values for different bacteria, and you take the bottom end of that distribution, and you're dividing that by 10, and that is actually how the PNEC concentrations are derived. Now, these may be quite misleading. In our view, they are quite misleading because this considers the MICs are derived from growth in rich media in the lab.
Yeah. Mm-hmm.
And that doesn't consider the fact that in the environment, growth rates are generally lower, and we have predators and phages and other nasty things in addition to the antimicrobials, and that does change the predicted selective window. And I think the PNEC values that are currently in use are not reasonable to use in the environment. And we're trying to do better.
Yeah, no, I guess, are any of the projects trying to estimate new PNECs, or I wasn't aware in our Indian one? We're not.
Yeah, I think Alan's project is trying to do that, but I don't know any details what he's trying to.
Is anybody from that project here available to comment? Laura, maybe Laura's here.
Yeah, I know Laura had one where she couldn't be here.
I had to come out because I had to give a talk halfway through, because I was meant to be in Germany, but it was all cancelled because of bloody COVID. So I had to dip out. So I might have missed a bit of your talk, which is, sorry for that, which is why I probably missed your.
No problem. I didn't have time to go into that anyway.
Yeah. Yeah, but yeah, I think this is, I know that Will's group are very busy trying to improve this situation. So, and there's several things, you know, it's kind of a complicated system where they're looking at single organism response as well as community response. So they also have this system where they have a community which is exposed, and then they get a minimal impact of that. And it can be a range of impacts as well, I think. Yeah, but that would be useful.
Mats, I wonder whether you had other slides or whether.
No, I don't actually have any more slides. I don't even have any conclusions or anything.
Yeah, that is probably wise. I mean, it was meant to introduce us to the procedure of, you know, what kind of information do you need, and how do you put that information together into evaluating risk. So thank you very much for doing that, Mats. If you do have any last minute questions, I'm conscious of time. We meant to conclude in a minute or so. If there are further questions, we can also collect them in the chat and try to answer. In the next session, we will reconvene Thursday at the same time, and then hear more experimentally oriented talks on Thursday.
I figured I might as well say something after having watched for the last few hours. I think what I was suggesting about mesocosm type experiments is useful, but there's another question that's part of the decision of what a PNEC actually means, and that is what is the risk? In other words, is the risk some sort of evolutionary development of new resistance? Is the risk related to the spread or transmission? And the value that might be of importance if you're establishing a value for a PNEC will differ completely depending upon what you perceive as your primary risk. And that's just one of the, I'm not a big fan of PNECs for AMR without, and I will go into my normal rant about that. But the bottom line is that it really, you really have to get your head around what you're really worried about. And it goes back to the beginning of what Matt said. And I'm hoping that maybe within some of the mesocosm experiments we're doing in the project Jan's leading, that we might be able to gather a little bit of information related to that, because we're doing smaller scale mesocosm studies there. But anyway, I couldn't keep my mouth shut after having kept my mouth shut for a few hours. But I really don't see any easy way of doing. I don't believe in lab studies. I think they're completely biased. You can get some information, but it's really the mixtures of the natural environment, both chemically and microbially, are just too complex to simulate that in the lab. And we failed many times trying to do that. So I fall back on what you and I did, Liz, about 15 years ago with mesocosms in central Kansas. And it really is the best way of doing it.
Yeah, they were proper mesocosms.
They're real. And in fact, Chuck Knapp and I have been talking about going back to our friends in Kansas, though they're both now here, and seeing if we can talk them into running a few more studies now that we've got all these more fancy ways of measuring things that we didn't have then. So
yeah, good idea.
Yeah, David, very good points. I think actually with the use of mathematical models, we can bridge the simpler laboratory experiments with the more realistic mesocosm experiments and measurements in the field, and this way deal with a problem that the more complex the system is, the more realistic it becomes, but the less you understand what's happening. And is a laboratory systems where you know what's happening, but it's not realistic, and really bringing all these studies at different levels of complexity together with models that can bridge the divide. That's kind of the vision that I have for this. So thank you all for attending this meeting. Please can I encourage all participants to feel free to ask questions anytime in the chat, or ask questions during the talks. It's meant to be workshops where we can have discussions and training of things that you may not be so familiar with. So with many thanks to our speakers Pranab and Mats today. Wish you a good day, and hope to meet you back on Thursday. So thank you all for your inputs and attending, and goodbye.